Chapter 18: Chronic Inflammation as Failed Completion. Inflammation is not defined by activation alone. It is defined by the ability to complete a sequence that begins with disruption and ends with restoration of structure. The initiating event alters membrane integrity, releases signaling substrates, and recruits cellular responses that address the disturbance. Completion requires that these responses terminate once the structural issue is resolved and that the membrane returns to a state capable of maintaining controlled separation. Chronic inflammation emerges when this sequence initiates correctly but fails to terminate, leaving the system in a sustained state of activation that continues to draw on resources without restoring baseline conditions. The process begins with structural disturbance at the membrane. Mechanical stress, chemical modification, or signaling overload alters lipid organization and triggers enzymatic release of fatty acids from phospholipids. These substrates are converted into signaling molecules that initiate local responses, increasing permeability, recruiting immune cells, and altering cellular behavior. This phase is necessary. It exposes the system to conditions that allow repair and containment of initial disruption. Completion requires a transition in substrate use and signaling output. The same membrane that provided substrates for initiation must also supply substrates that guide resolution. This involves shifting from signals that promote activation to those that reduce it, clear damaged components, and restore membrane organization. If the membrane lacks the appropriate composition to support this transition, signaling remains biased toward continued activation. The system continues to operate in a state designed for response rather than restoration. Structural conditions determine whether this transition can occur. Membranes with sufficient organization and balanced lipid composition provide both the stability required to contain activation and the substrates needed for termination. When structure is compromised, permeability remains elevated and signaling pathways continue to be engaged. This prevents the system from re-establishing the conditions required for completion, extending the active phase indefinitely. Persistent activation alters cellular behavior across multiple systems. Immune cells remain engaged, producing signals that recruit additional cells and maintain the response. Endothelial membranes increase permeability, allowing continued movement of cells and molecules into affected areas. Signaling pathways remain active, altering metabolic and hormonal regulation. These changes do not resolve the initial disturbance. They sustain the response in the absence of completion. Energy demand increases under these conditions. Maintaining an active response requires continuous production of signaling molecules, transport of substrates, and repair of ongoing damage. At the same time, structural instability increases leakage and inefficiency, requiring additional energy to maintain gradients and basic cellular functions. The system operates under sustained load, reducing its capacity to complete the processes required for resolution. Remodeling cycles are also affected. Persistent activation increases the rate of lipid turnover, but if the available substrates do not support stable reconstruction, the membrane remains in a state of altered composition. This prevents restoration of the structural conditions required for termination, reinforcing the cycle of continued activation. Chronic inflammation, therefore, represents a failure of completion rather than an excess of initiation. The system begins a response correctly, but cannot transition to termination because the structural and compositional conditions required for that transition are not present. Activation persists, resources are consumed, and the membrane remains in a state that continues to generate signals rather than resolve them because the architecture required to close the loop has not been restored. Persistent signaling loops. Persistent signaling loops form when activation no longer depends on an external input and instead sustains itself through internal structural conditions. The membrane is the surface where this shift occurs. When lipid organization becomes irregular and permeability increases, signaling components encounter one another more frequently and remain associated longer than intended. What begins as a response to a discrete trigger transitions into a self-maintaining cycle where the output of the system feeds back into its own activation pathways. The loop begins with incomplete termination. Signaling molecules are generated and propagate through receptors and downstream pathways, but the mechanisms that normally reduce their concentration and separate interacting components do not complete their function. Lipid-derived mediators continue to be produced because the substrates remain available and the structural conditions that would shift production toward resolution are not established. Receptors remain in proximity, channels continue to cycle, and the membrane does not return to a baseline state that would reduce interaction frequency. Structural permeability reinforces this process. Increased ion leakage alters electrical gradients, which changes the behavior of ion channels and transporters. These changes influence intracellular conditions, maintaining a state that favors continued activation. The membrane no longer isolates the internal environment from external variation. Instead, it allows fluctuations that sustain signaling activity even in the absence of new stimuli. Reactive byproducts contribute to loop persistence. Oxidative modification of lipids generates molecules that act as additional signals, recruiting further activity and altering cellular behavior. These signals originate from the same structural instability that prevents termination, creating a feedback relationship where damage and signaling amplify one another. The membrane becomes both the source and the amplifier of the loop. Immune involvement extends the cycle across systems. Persistent signals recruit immune cells that release additional mediators, increasing local activity and altering surrounding tissue behavior. These inputs do not resolve the original disturbance. They add new layers of signaling that maintain the loop. The boundary between local and systemic response begins to blur as signals propagate beyond their initial location. Energy demand increases as the loop continues. Maintaining gradients, producing signaling molecules, and attempting repair all require ADP. As structural instability increases leakage and inefficiency, more energy is required to sustain basic conditions. This reduces the system's capacity to complete termination processes, further reinforcing persistence. Remodeling cycles fail to break the loop when input composition remains unchanged. As lipids are replaced, the same structural conditions are reintroduced, preventing restoration of a stable baseline. The membrane continues to support conditions that favor interaction and signaling rather than separation and termination. Persistent signaling loops therefore arise when structural conditions prevent the system from exiting an activated state. Increased mobility, permeability, and reactive byproducts maintain interactions that should be transient. The loop sustains itself through feedback between structure and signaling, extending activation beyond its functional purpose because the membrane no longer enforces the boundaries required to return the system to baseline. Inadequate resolution substrate availability. Resolution requires specific substrates embedded within the membrane that can be released and converted into mediators that terminate active responses. These substrates must already exist within the phospholipid structure at the moment activation occurs. If they are absent or present in insufficient proportion, the system cannot generate the signals required to shift from activation to termination. The membrane initiates the response using available components, but lacks the material needed to complete it. This imbalance begins with composition. Membranes enriched in substrates that favor initiation will generate signals that recruit, amplify, and sustain activity. If the proportion of omega-3 derived substrates is low, the pathways that produce resolving mediators are undersupplied. Enzymatic systems that would normally convert these substrates into termination signals remain active but underutilized because the required inputs are not present in sufficient quantity. The system continues to produce initiation signals from dominant substrates, reinforcing the active state. Availability is not determined at the moment of signaling. It is set during prior remodeling cycles that establish membrane composition. Each cycle determines which fatty acids are embedded within phospholipids and therefore accessible for release. When these cycles do not incorporate sufficient resolution-supporting substrates, the membrane enters activation phases without the capacity to generate corresponding termination signals. This creates a structural bias toward persistence. Spatial distribution further limits availability. Even when resolution substrates are present, they must be located within the regions where signaling occurs. If they are unevenly distributed, localized areas of activation may lack access to the necessary components, preventing resolution in those regions. This creates heterogeneity within the membrane, where some areas complete responses while others remain active. Competition between substrates intensifies this limitation. Enzymatic pathways that process fatty acids can act on multiple substrates, but they do not produce the same outputs from each. When initiation-favoring substrates dominate, they occupy enzymatic capacity, reducing the conversion of resolution-supporting substrates even if they are present. The balance of inputs determines which pathways are favored, and this balance is set by membrane composition rather than immediate demand. Turnover and replenishment influence long-term capacity. Resolution substrates that are used during signaling must be replaced through remodeling cycles to maintain availability for future events. If replenishment is insufficient, each cycle of activation reduces the system's ability to complete subsequent responses. Over time, this leads to a progressive decline in resolution capacity. Energy availability constrains these processes. Synthesis, incorporation, and enzymatic conversion all require ADP. When energy supply is limited, the system prioritizes immediate survival functions, reducing the resources available for maintaining substrate balance. This further limits the ability to generate resolution signals when needed. Inadequate resolution substrate availability, therefore, reflects a structural limitation embedded within the membrane. The system can initiate responses but cannot complete them because the components required for termination are not present in sufficient quantity or appropriate location. This imbalance sustains activation, increases resource demand, and prevents restoration of baseline conditions because the membrane lacks the materials needed to close the signaling sequence it has begun. Immune system misalignment. Immune function depends on the ability to distinguish between what should be engaged and what should be ignored, then to terminate that engagement once the task is complete. This discrimination is executed at membrane interfaces where receptors detect signals, assemble into complexes, and initiate downstream responses. When membrane structure drifts away from controlled organization, the conditions that govern recognition, activation, and termination shift. The immune system does not lose its components. It loses alignment between input and response. Recognition is the first point of failure. Immune receptors rely on spatial organization within the membrane to interpret signals correctly. Clustering must occur within defined domains to confirm that a stimulus is sufficient to warrant activation. When membrane fluidity increases beyond controlled limits, receptors encounter one another more frequently and outside of structured regions. This lowers activation thresholds and increases the likelihood of engagement in response to weak or ambiguous signals. At the same time, irregular packing alters how ligands interact with receptors, reducing the precision of detection. The system begins to respond to inputs that would normally be filtered out. Activation follows this altered recognition. Once engaged, signaling pathways amplify the response through coordinated interactions between membrane-bound and intracellular components. Structural inconsistency affects how these interactions propagate. Some regions amplify signals excessively due to increased mobility, while others fail to sustain them due to lack of stable clustering. The result is uneven activation, where the magnitude and distribution of the response no longer reflect the original stimulus. Termination is impaired under these conditions. Resolution requires separation of signaling complexes and the generation of mediators that reduce activity and restore baseline conditions. When membrane structure does not support controlled disassembly, signaling components remain associated longer than intended. At the same time, inadequate availability of resolution substrates limits the production of signals that would normally guide termination. The response persists even after the initial trigger is no longer present. Boundary function becomes less distinct. Immune cells depend on membrane integrity to maintain separation between internal and external environments, as well as between self and non-self. Increased permeability and structural irregularity blur these boundaries, allowing signals and molecules to cross in ways that alter how cells interpret their surroundings. This can lead to engagement with targets that should remain ignored or failure to respond appropriately to actual threats. Communication between immune cells is also affected. Cytokine signaling and cell-to-cell interactions rely on consistent membrane behavior to transmit information accurately. When structure varies, the same signal can produce different responses depending on local conditions. This reduces coordination across the immune network, leading to responses that are either exaggerated or insufficient. Energy demand increases as the system attempts to maintain activity under unstable conditions. Persistent signaling, increased transport activity, and ongoing repair processes require continuous ADP input. This diverts resources from resolution and restoration, reinforcing the state of misalignment. Remodeling cycles influence whether alignment can be restored. If membrane composition shifts toward a structure that supports controlled packing and balanced fluidity, receptor behavior and signaling dynamics return to a more precise state. If destabilizing inputs persist, remodeling reinforces the same conditions that drive misalignment, preventing correction. Immune system misalignment therefore arises from structural conditions that alter how signals are detected, processed, and terminated. The system remains active, but its responses no longer correspond accurately to input because the membrane environment that governs interaction and interpretation has lost the consistency required for precise discrimination and controlled completion. Tissue-level consequences. When membrane structure shifts across a population of cells, the effects no longer remain confined to individual signaling events. They scale into tissue behavior because every cell within that environment operates under the same altered constraints. The membrane defines permeability, signaling precision, and energy efficiency at the cellular level. When these properties drift, the coordination between cells begins to degrade, and the tissue loses its ability to maintain stable function under load. Barrier tissues are affected first because they depend directly on controlled permeability. Epithelial and endothelial layers rely on tightly regulated membranes to maintain separation between compartments. When packing density decreases and permeability increases, ions, water, and signaling molecules cross more freely. This alters pressure gradients and fluid balance, leading to leakage and swelling within the tissue. Junctional integrity becomes harder to maintain because the underlying membrane no longer supports consistent alignment. The barrier becomes inconsistent rather than uniformly compromised, with regions of higher and lower permeability that disrupt overall function. Signal coordination between cells becomes less reliable. In a stable system, signals propagate in a controlled manner, allowing groups of cells to respond together. When membrane properties vary, identical signals produce different responses depending on local conditions. Some cells activate rapidly, others respond weakly, and some remain active longer than intended. This desynchronization disrupts tissue-level processes that depend on timing, such as coordinated contraction, secretion, or immune response. The tissue begins to operate with internal variability rather than unified output. Energy demand increases across the tissue as each cell compensates for increased leakage and signaling noise. Maintaining ion gradients requires more active transport, and persistent signaling loops consume additional resources. Cells allocate more energy toward maintaining basic conditions and less toward specialized functions. Over time, this reduces the overall efficiency of the tissue, limiting its ability to perform its primary role. Structural integrity declines under mechanical stress. Tissues experience continuous forces from movement, pressure, and flow. When individual cell membranes cannot distribute these forces effectively, stress concentrates at points of weakness. This leads to micro-level disruptions that accumulate, altering tissue architecture. Cells may detach, deform, or fail to maintain proper orientation, affecting how the tissue responds to load and how signals are transmitted across it. Inflammatory signaling becomes embedded within the tissue environment. Persistent activation at the cellular level leads to continuous release of mediators that affect neighboring cells. This creates a local environment where signaling remains elevated even in the absence of an external trigger. The tissue shifts toward a state where baseline conditions include ongoing activation, altering how it responds to new inputs. Repair processes become less effective. Tissue maintenance depends on the ability to remove damaged components and replace them with functional ones. When membrane instability persists, newly formed cells inherit the same structural conditions, preventing full restoration. Remodeling cycles operate, but they reproduce the existing state rather than correcting it, embedding instability into the tissue over time. Communication with other systems becomes distorted. Hormonal, neural, and immune signals entering the tissue are interpreted through membranes that no longer provide consistent conditions. This alters how external inputs are translated into cellular responses, affecting integration with broader physiological systems. The tissue no longer responds predictably to systemic regulation. Tissue-level consequences therefore reflect the aggregation of cellular instability into a larger system. Variability in permeability, signaling, and energy use spreads across the cellular population, disrupting coordination and reducing functional capacity. The tissue remains active, but its behavior becomes less precise and more energetically costly because the structural conditions that allow cells to operate as a coordinated unit are no longer maintained.